Hi, everyone.
Hi, how are you?
I'm well. How are you today?
I'm good.
Good. Glad to hear it. Nice day. Nice and warm today.
Yeah. I'm still in isolation, so
Oh, you are? Oh,
but soon I get out tomorrow, so
well, that's good. I hope you're feeling better.
I am, thank you.
That's good. I hear that tomorrow is supposed to be a pretty nice day too. So maybe spring is on the way.
Hopefully, fingers crossed.
I'm always hesitant to say that. I'm always afraid that if I say it, then it'll snow or something, But who knows? Maybe we'll get some warm weather soon.
Hopefully.
All right. So it's just a minute before 4 o'clock. I'm a little concerned that more people aren't here. I'm just rearranging some things here. I'm just checking to make sure our link is up. All right, let's see here. I don't see any messages from anybody having trouble getting in, which is good. So I'm going to just go ahead and get started. I hope everybody's doing well. I hope things are going smoothly for you. I'm just going to pull up for us our schedule for just a minute so we can take a look at what's coming up next week. So I'm just pulling up a screen that shows our Canvas module page, and I'm just scrolling down. This is where we are now, of course. We're in the week of March 8th, and this week's lecture material includes both the eukaryotes and acellular microbes, so the viruses and other particles that can cause disease in humans. And today in lab, we're going to be talking about testing for microbes in our environment. Now, looking ahead to next week, so on Tuesday next, you have your second lecture exam. And remember, this exam is going to cover lecture material since the first exam. So it will cover the prokaryotes topic, the eukaryotes, and the acellular microbes. So everything that you've gone through since the first lecture exam. It'll look just like the first lecture exam in terms of the time that you can take it. You're allowed to have your notes with you during the exam. It'll be due at 8 p.m. unless other arrangements have been made. So that's coming up on Tuesday next week. Now, in terms of lecture material next week, the topic is microbial biochemistry. Now, this is, in my opinion, one of the hardest topics that we have in this course. So just be aware that this is coming up next week, and it may just require a little extra time on your part to go through the lecture videos and make sure you have complete set of notes for yourself. Some people find chemistry and chemistry related topics very easy, and other people find them more challenging. So just be aware that there's this topic coming up that might need a little extra care from you. There'll be that one lecture quiz next week for microbial biochemistry. And we will have a Zoom meeting on Tuesday. Okay, so you have a lecture exam on Tuesday. You also have lab on Tuesday. Okay, and our topic for next week in terms of the lab is special media. Now, when I use that phrase, special media, I'm talking about a specific class of culture media. We've talked about culture media that can grow just about anything. We talked about nutrient agar, for example, as being a type of media that you can grow almost anything in because it has some basic nutrients in it that microbes need to grow. But we also have an entire collection of different kinds of media that we use very specifically to promote the growth of one type of microbe while inhibiting the growth of another kind of microbe. For example, we have media where only gram positives will be able to grow and all gram negatives will not be able to survive on the media, and vice versa. We have media that can help us distinguish different strains of bacteria. So that's what we refer to very generally as special media. We're using them for very specific purposes to grow very specific types of organisms. So that'll be coming up next week. If you have any questions between now and next Tuesday regarding any of the lecture material that you're viewing, feel free to message me. And we can talk through that together. Hopefully everything is pretty straightforward for you, but always feel free to connect with me if you need to go over any of the lecture material. I'm happy to do that with you. All right. So any questions? Any questions about any of the materials that you've been looking at, the lecture materials you've been watching, any of the quizzes you might have taken? Any questions at all? You can type them into chat if you'd like. And certainly as we go through class today, if anything comes to mind, you can certainly send me a message on chat. You can also message me after class. All right, I'm going to pull up our lecture slides now, or I should say our lab slides now, for this topic called microbes in the environment. And that's what you should be looking at right now, is a slide that says lab, microbes in the environment. So our objectives today for this lab exercise are to talk about the different microbial testing methods that we have available for us with regards to monitoring the kinds of microbes that grow in various environments. We're also going to talk a little bit more about culture media. We're going to talk about Auger today and about why it's such a nice choice as a solid culture media. We'll compare the way we use Auger in the microbiology lab. Obviously we can pour it into Petri dishes and make Auger plates. That's one way we use it. But we can also pour it into glass test tubes in a couple of different ways and use that auger for different purposes. So we'll talk about that. And we'll talk a little bit about the differences between how bacteria grow in a liquid broth versus a solid agar medium. And then lastly, we'll talk just a little bit about colony morphology. So, a little bit of background first about monitoring microbes in our world, in the environments that we are in each and every day. Remember, as we've said in here before, and as you'll hear me say again, microbes are everywhere. They're always around us. Most of the microbes that we are surrounded by every day, whether they're on our bodies or they're in our the kinds of environments that we find ourselves in, most of them are perfectly harmless. In fact, a lot of them are quite helpful to us, as we've talked about, but a few of them are pathogens. So there are occasions where we need to monitor for these pathogens. We need to know if they're present in the environment, and also if they are, what numbers of them are present in the environment. So sometimes we're monitoring natural environments. Sometimes we're monitoring man-made environments. So by natural environments, things like bodies of water, recreational bodies of water in a place like New Hampshire get regularly monitored for the growth of pathogenic microbes, because we want to be sure that people who are making use of these water bodies, people who are tourists or who are residents that are maybe water skiing or swimming or fishing, that they're going to be safe. Water treatment plants are a man-made kind of environment where we have to monitor different types of microbe levels, because we want to make sure that the treatment process is functioning normally. Public water systems. So a municipal water delivery system that is sending water into people's homes, that has to be monitored. And of course, places like hospitals, doctors' offices, long-term care facilities, even places like industrial clean rooms, the kind of places where industrial products are made that have to be made in a place where it's not going to be possible for dust or cells or any kind of spores to fall onto the materials. There are lots of places where clean rooms or sterile rooms are maintained in order to protect the product that's being made, essentially. So all of those places have to have programs in place that allow for this monitoring process. They have to know if there are microbes growing, and if they are growing, what species are they? Are they pathogenic? And to what extent are they growing? How many of them are growing in that moment in time? All right. All right, I'm just going to go back here now. So essentially, when we're talking about these monitoring programs, we're talking about people being assigned this task of taking samples from these environments and then sending the samples to the microbiology lab so we can make an effort to determine whether or not microbes are present. And if they are present, to try to identify them. Sometimes we are sampling surfaces. And generally speaking, when we're sampling just dry, hard surfaces, we can use something like a swab in order to sample that surface, in order to collect any microscopic cells that might be sitting on that surface. And again, when that swab gets sent to the microbiology lab, we would use that swab to then inoculate some culture media and see if we grow anything. Sometimes we're not sampling a surface, a hard surface, so much as we're sampling, say, an air duct or some kind of forced air delivery system. This picture down here is pretty gross. It's a pretty filthy vent cover here. But the idea is that, microbes can grow anywhere. And it's certainly possible for microbes to grow in ventilation systems, again, in office buildings and in hospitals and doctors' offices and all kinds of different places. And if the ventilation system isn't working correctly, if enough moisture is around and there is not enough airflow, then microbes can certainly grow in ductwork. And that would be dangerous because when the forced air passes through the duct, it certainly at least would be possible for cells to be carried in the air and sent down into office spaces or procedure rooms in a hospital and so on. So those have to be monitored. Over here, this surface, this could be any kind of a surface in a medical facility. It might be a surface in, let's say, a cafeteria space or other food handling space. Sometimes we are literally sampling places that have been cleaned in order to make sure that they were cleaned correctly. That, for example, this floor, maybe this is in a laboratory space or some other kind of a research facility. We want to make sure that whatever procedures we're using to clean this room, that we're successful in cleaning that room and we're not leaving a lot of microbes behind. Now, of course, there are also times when we have to collect liquids. Remember, I said one of the places that we sample, particularly in New Hampshire and the rest of New England, are bodies of water. So it's very common for us to gather water samples in order to do this. And because those samples tend to be very, very dilute, in other words, if there are microbes out in that lake that people are swimming in, the chances are there are very few microbes for the entire volume of the lake. So when we collect liquid for evaluation, we often have to run that liquid through a centrifuge in order to collect the cells, in order to sort of harvest the cells out of that liquid. And then again, we can inoculate some media with those cells when they arrive in the laboratory. It's important that the folks who gather samples from these different environments know what they're doing. In other words, they have to know aseptic technique. They have to know how to collect the sample. They have to know where to collect the sample. And they need to know what to do once they have the sample in order to get it back to the micro lab without being contaminated. On this slide, what you're looking at are a couple of commercially available swabs. You can see each swab has a color-coded plastic handle, and then there's this, hard to see, but there's a white swab with cotton at the end of it. And again, you could use a swab like this by swiping it over a surface that you want to test, that you want to monitor to see if there are microbes growing. After you collect cells on a swab, or at least potentially collect cells on a swab, you have to get them to the laboratory alive. And microbial cells are not going to survive for very long on a dry cotton swab. So these swabs have to be placed back into their containers. That's what this is right here. There is media in this container. There's what we call transport media. And when the swab is placed back in here and the container is closed up, that transport media is going to allow those cells, if they're present, to stay alive during the journey to the microbiology lab. Transport media is designed just to keep the cells alive long enough so that they can get to the laboratory for evaluation. And there are lots of different products available in terms of swabs and their containers, but depending on the purpose, depending on what you expect you might be getting on your swab. There are different swab types. There are different types of transport media. Um, all of this stuff is available commercially now. On this slide, you're looking at several images of people collecting water samples. For example, these two women here are collecting water samples from a swimming area in a lake. Um, you can see that this woman has picked up sort of a pitcher full of water, and she's taking a sample in a bottle. Now, these bottles, I have an image over here on the lower right-hand side. These bottles come in all shapes and sizes, again, commercially available. They're sterile, of course, before you open the lid anyway, they're sterile. And you can collect different volumes of fluid depending on the circumstance, the environment you're trying to monitor. So I think you can just see this buoy line right here. This is likely a swimming area that has been roped off for this lake, and it's likely that these women are collecting to monitor for pathogens that might be growing in the water in the swimming area. Now, you can't just take a single sample to do this. You can't just go out and draw up 50 or 100 mLs from the middle of the swimming area and call it a day, because you want to be able to say that the whole swimming area is safe, right? You want to be able to say that. So you have to collect several samples, what we would refer to as a representative sampling. You might want to collect a couple samples from the far corners of the swimming area. You would probably want to collect a couple of samples from up near the beach, up near the sand. You'd want to make sure you got samples all along the length. Um, sorry, I'm just reading a message. Um, you'll want to make sure that you collect enough samples so that you feel like you've got the entirety of that swimming area represented. Um, this is true anytime that you're trying to monitor a large area. So if I'm trying to, for example, determine whether or not a water treatment facility is functioning correctly, I can't draw a single sample out of that facility and know enough. I'm going to have to take lots of samples from different places in that facility. If I want to know what's happening with a private well, here in New Hampshire, a lot of people use, backyard wells for their drinking water, and you'll want to make sure that you get samples from a couple of different places in the well. You have to make sure that you're getting enough material so that it's representative of that entire body of water. All right. Now, on this slide, what you're looking at are some meters that are available commercially now at a lot of different laboratory supply type places. These are meters that are designed to monitor the air. Now, typically, if we're trying to get a feeling for microbes that are present in an air system, let's say a ventilation system, we can take swabs inside of ductwork. We can take swabs from grates that are on the opening of ducts. But we can also get samples from the air itself. And you can do that by placing filter material in the path of the airflow. So the air flows through the filter. And as long as you use a filter that has the appropriate pore size, you can trap bacteria and other types of microbial cells in the filter, and then use the filter then to inoculate culture media when it gets to the lab. But these devices, These devices are capable of sampling the air without a filter. There's a filter inside of here, but they're capable of sampling the air specifically for spores. So, what this kind of device is doing is it's generating a spore count per some volume of air. Now, I wanted to show you these because there is a bit of a problem with these devices that you have to know about. You have to know the limitations of the equipment that you use. It's possible now for people who own homes to hire private companies to come into their homes and tell them how many spores are in their home, again, per volume of air. So instead of collecting a sample, say with a filter, and then sending that sample to the laboratory for analysis, basically what these companies do is they come in and they tell the homeowner, you have, say, 100 spores per centimeter cubed of air. Um, there's a real problem with this in that, well, there's a couple of problems. The primary problem is that just because this device can measure a spore, it doesn't know what kind of spore it is. So remember, we have talked about in here how bacteria, certain types of bacteria anyway, are capable of sporulating, right? We said the Bacillus genus and the Clostridium genus are capable of sporulating under certain conditions. They go from a vegetative state or a cellular state, and they become these inanimate spores. It's basically a little hard container of DNA that is resistant to all kinds of forces that might kill it. Doesn't metabolize, it doesn't need any nutrients, it doesn't generate any wastes. It just sort of sits there until the environmental conditions are once again good enough to support a living cell. And then spores can revert back into their vegetative or cellular forms. And spores do get into homes. I mean, there are lots of non-pathogenic spores of bacteria. They're in the soil. They can be found in dust. Certainly in older homes, you'll find spores of bacteria. Fungi also sporulate. So fungal spores are very, very common in homes, in residences. But again, what this meter is measuring is just the count of spores, and that's all. And you can imagine that a lot of homeowners get very frightened when they hear that they have, 50,000 spores in their home. Um, so it's not really a great practice to stop with that count because you're not telling anyone what kind of spores they are. It's very normal, it's very natural for there to be fungus in a home, especially in an older home. Here in New England, a lot of the homes that people live in are old, sometimes hundreds of years old, right? And all that old wood and old insulation can harbor a lot of fungal spores. But those fungi are not pathogens, so they're not a risk to the homeowner. And the fact that there are spores there instead of actual cells tells you that, the home in those areas is actually nice and dry. And there aren't the kinds of nutrients and water that are necessary for these cells to come back, and they stay as spores and they're not really dangerous to anyone. So it's unfortunate that this industry has sort of grown out of fear. And a lot of homeowners will, they'll hear that there are, again, thousands of fungal spores in their basement or something, and they get really frightened and they feel like, If they're looking to buy the house, they might think they shouldn't, or they may think that they have to tear their house up and treat. That's not necessarily true. Anytime we're monitoring, the presence of a spore or a cell doesn't necessarily tell the whole story, right? What we really need to know is what kind of an organism is there. If there are fungal spores, what kind of fungus is that? And is it a pathogen? Is it a danger to anyone? So you have to be careful. You have to know sometimes when you're hiring these companies, you have to know what exactly this information means that they're giving you, and how much you can trust it, to make decisions based off of, basically. All right. So I want to switch gears here just a little bit. I want to talk about a very particular tool that is used in healthcare settings primarily to monitor environments for bacteria, and also fungi, but primarily bacteria. And this is a device called a Rhodac plate. Now, the term Rhodac refers to replicate organism detection and counting. And it looks like this. Now, it looks almost like the bottom of a petri dish in that, it's a plastic container and it's got agar in the bottom of it. This person is holding this plate upside down. So the agar is actually exposed right here. So what you do with these plates, the plastic that they're made out of is actually very flexible. Unlike the plates that we use in the microbiology lab, these are made out of a soft and flexible plastic. And they come with a lid on them. What you do is you take the lid off, you turn them upside down, and you press on the bottom of the plate so that the agar actually sort of bulges out. And then you press that agar down onto surfaces so that if there are any cells on that surface, you will pick them up on the agar. So this person has taken the lid off, flipped the plate upside down, and is pushing with her finger on the bottom of the plate to cause the plate to bend so that the agar surface inside that plate is now sort of jutting out. And then you literally just press it down onto the surface a couple of times, put the cover back on, and send it to the laboratory. So essentially what the Rhodac plate does is it allows you to skip a couple of steps. Instead of taking a swab like this pen and rubbing it on a surface and then sending the swab to the lab and having the technician in the lab then take the swab and put it on an agar plate. Instead of doing all those steps, the person who's collecting the sample is actually putting cells onto agar. So you take those plates after you've pressed them on a surface, you put the cover on, and they go into the incubator. Some hospitals will send the Rhodac plates to a microbiology lab so that the plates can be incubated at the lab and evaluated at the lab. But some hospitals just keep those plates in-house and they have their own incubator. And they'll take samples with the Rhodac plates and then they'll stack them in their incubator. They let them incubate for about 24 hours and then they evaluate them. So Rhodac plates are nice because they're easy to use and the several steps have been eliminated in terms of determining whether or not there are microbes growing. Now, as I said before, if you were in a hospital setting and it was your job to, say, perform monthly or quarterly sampling of different places in the hospital to monitor for microbes, you would never just take one sample. If, for example, you were sampling, let's say, an operating room, you would want to probably take 10 or 20 Rhodac plates into the operating room, and you would want to press the plate onto all different kinds of surfaces in the room, so that you get a nice representative sampling of all the different surfaces in the operating room. So you can look for potential pathogens that are growing in that room. Now, on this slide, we're looking at a couple of agar plates, regular agar plates, that have bacteria growing on them. This plate over here on the left, this is a plate that was created through quadrant streak plating. And this plate over on the right was created using what's called a spread plate technique. So we can put bacterial cells onto agar a couple of different ways, depending on what we're trying to achieve. Now, when a Rhodac plate gets put into the incubator and we evaluate it the next day, there's a good chance that if there are cells on this surface, what we're going to get growing on this plate are little individual colonies of growth. So on this plate over here on the right, what you're looking at is hundreds of individual colonies of growth. So individual cells landed on this agar, were actually spread out on this agar, and then grew into these circular colonies overnight. So each colony is actually a clump of cells. The original cell that landed at that spot on the agar, and then all of the daughter cells, the progeny cells or the offspring cells of that original cell. If that cell is alive and healthy, it's going to divide again and again and again and again overnight as it sits in the incubator. And by morning, you'll have colonies growing. So when we make a spread plate like this one, we take a volume of culture material. We take a small volume of material out of a broth tube, and we put it in the middle of the agar plate. And then we use that hockey stick device to spread that material out very evenly over the entire surface of the agar. This is what we call, again, spread plating. We're spreading all the cells out. We want to take advantage of all of this surface area. And the reason we make a spread plate like this is because we're going to count these colonies. We're going to count the colonies. And after we do a little bit of math, we're going to get an estimate of how many cells per volume of material were in that original tube. So spread plating is used in order to count colonies and come up with an estimate of cell number in a particular sample. A quadrant streak plate is very different. So this is a quadrant streak plate, and like the name suggests, what we do is we separate the plate into quadrants, and we streak the quadrants in a very particular way. So you have to imagine that there's a line drawn across here horizontally and another one drawn vertically to split this plate into 4 separate compartments, basically. And what we do is we take a loop full of broth culture and we just spread it out into the first quadrant. We just spread all those cells into the first quadrant. And you're going to get growth that looks a little bit like this. Everywhere you see this cream colored sort of material growing, these are many, many, many bacterial cells growing. Now, the purpose of creating a quadrant streak plate is to take all of these cells and manipulate them in a way that will allow us to grow individual colonies. There are many cells that come out of a broth tube, a broth culture tube. You can see that because when we smeared that material onto the plate, it grew as this big stripe. So how do I go from there to grow individual colonies? Well, I will sterilize my loop. I'll draw it through that smear. Actually, I won't be able to see the smear yet because this is while we're making the plate. I will draw it through the location that I just streaked into the next quadrant, and I will streak again. Then I sterilize my loop. I draw it through that second quadrant, picking up a few cells, and then I streak again. And then I do it a fourth time. I sterilize my loop. I draw it through the third quadrant, and I streak again. And hopefully in maybe the third or perhaps the fourth quadrant, I will have reduced the number of cells to the point where I can get individual colonies to grow. Now, the purpose of this type of quadrant streak plate is to get these colonies not for counting purposes, not for counting, but just to describe the colonies. This is what we refer to as colony morphology. Remember, that word morphology translates as body form. And it turns out that different kinds of bacteria, different species of bacteria, grow into colonies that have a unique appearance. They can have different colors, they can have different sizes, they can have different shapes. They can rise up above the auger to different heights. There are lots of characteristics of these little clumps of cells that we call colonies that are unique to that organism. And we refer to that as the colony morphology for that organism. Now, there are limitations to colony morphology because it is possible for more than one species of bacteria to have an identical colony morphology. The analogy I like to use is that it's very similar to human beings having the same hair color and the same eye color. You're two different people, but you share certain characteristics in common with each other. Bacteria are the same way. So, if I see colonies growing on a plate and they're all 2 millimeters wide and they're circular and they're cream colored and they're glistening and they have a nice rounded convex surface, that might be the colony morphology for 24 different bacteria. So I can't use colony morphology to necessarily identify a bacterium, but it is a piece of information about them. So if I want to evaluate the colony morphology for a bacterial species, I will make a quadrant streak plate. If I'm instead monitoring an environment by doing some sampling, I'm going to make a spread plate because my purpose is to count those colonies and then do some math to figure out how many microbial cells there were in that 500 mLs of water or in that 60 centimeter squared area. So spread plating is about determining microbe numbers, and quadrant streak plating is about looking at colony morphology. We're actually going to talk a little bit more about how we make spread plates later in the semester. But when we take these Rhodac plates out of the incubator, we haven't spread anything on these, have we? We haven't done a quadrant streak. We haven't done a hockey stick spread. We haven't done any of that. We've just pressed them onto the surface. So depending on what you're sampling, you may get some individual colonies growing. You may get a big clump of bacteria growing. But what we're going to try to do is count colonies on these Rhodac plates. So what exactly is this colony, this clump that's growing on Auger? Whether it's on a Rhodac plate or a quadrant streak plate or a spread plate, what exactly is a colony of bacteria? Well, we define it as a visible, that's important, a visible mass of bacterial clones that all came from a single cell on that solid culture media, that agar. So whether we put it onto an agar plate in the microbiology lab, or whether we picked it up by pressing a Rhodac plate down onto a surface, if you pick up a parent cell on agar and you put that cell into the incubator on that agar, that cell is going to divide. And as it divides, every time it divides, it makes two brand new cells, two daughter cells. And then those two daughter cells are going to divide. And you're going to have 4 cells. And then they're going to divide. And you're going to get 8 cells and 16 cells and 32 cells. And in a 24-hour period, that kind of exponential growth is going to give you hundreds and hundreds of cells in that colony. Now, that term clone is important because bacteria, when bacteria grow, they divide asexually. So that parent cell is going to create 2 new daughter cells through an asexual process. And what that means is the daughter cells are going to be genetically identical to the parent cell. So we refer to them as clones. So every single cell that is in a colony is genetically identical to the parent cell, that original cell that landed on the agar in that spot. Every colony is a collection of clones. They're genetically identical cells. So when we're monitoring microbes in the environment, remember, we're trying to figure out how many cells are in a particular place. It might be per surface area, it might be per volume. With the Rhodac plates, we're simply going to count what we call colony forming units, or CFUs, on the Rhodac plate. We can compare that count to standard reference tables to get an idea whether or not that particular environment is being disinfected correctly. How many cells are too many cells in an operating room, or in a patient's room in a hospital, or in a procedural room, or in a cafeteria, or at that water treatment plant? How many are too many cells? There are reference tables available for us to look at to make that determination. When we count colonies, if you think about it, we're actually getting an estimate of the number of original cells in that sample. We can't call it cells. We have to refer to it as colony forming units because it's not a direct measure, it's an indirect measure. But by determining how many colony forming units are on an agar plate, we can then extrapolate to know how many cells were in an original sample. So by growing cells on agar, we can get an idea of what's growing in that recreational lake, in terms of numbers anyway, not the identity of the organism. We can get an idea of what's growing in that hospital or what's growing in the doctor's office. We can get an idea of how effective we are in our cleaning and disinfection procedures. We can tell whether or not that water is safe for people to swim in. All right. Does anybody have any questions about what I've been talking about so far? Does it make sense why we bother looking at these commercial spaces and these recreational spaces and healthcare spaces? Why we want to know what's growing in these spaces? We expect there to be microbes around us in the world all the time. They're everywhere. They're all over us, they're all in us, and they're all around us. They're in our homes, they're in our workplaces, they're outside in the natural world. It's important to remember that most of them aren't going to cause any problems for us, but there are pathogens. And so it's important that we monitor these spaces so that we know, are there pathogens growing here? And if there are, how can we reduce their number? You know, what can we do to better control what's growing in these spaces that we need to be safe? You know, and again, it depends on what space you're monitoring, but by doing this kind of sampling and then going into the microbiology lab and using things like spread plating, we can determine at least a good estimate of what we have around us and whether or not we need to take steps to reduce the amount of microbes that are growing. There are even programs in food production facilities where they simply take samples of the food and they spread it out on an agar plate. And they, again, incubate that plate and count colonies, count colony forming units, in order to get a sense of whether or not they've got bad contamination in their food production system. This kind of monitoring takes place all the time in lots of different facilities and different kinds of environments. All right. I'm going to jump back in and talk just a little bit more about these colonies and colony forming units. So again, you have to think, you have to imagine in your head what happens when I take, for example, a loop full of broth out of a culture tube, a tube that I've been growing microbes in. I take a loop full of that liquid out and I spread it across a plate. Let's say I use a hockey stick. So I'm going to spread that broth all over every inch of that plate, and I'm going to spread it evenly across the plate. When I put the plate in the incubator, I'm not going to see anything on the plate, right? Because the cells will be present on the agar, but they're still microscopic. They're invisible to me. But after I put that plate in the incubator for 24 hours, everywhere a cell landed on the agar, I should see a colony now, a visible mass of cells that all came from that first cell that landed in that spot. That parent cell divides and divides and divides and divides overnight, creating this little visible mound of cells. So I've got a lot more cells by morning than I originally had, but I can count those colonies, calling them colony forming units again. I could do some math, and I can then say with some assurity, at least a good estimate of how many cells were per volume were in that original broth tube. So take a look at this slide here. There's a couple images here. This is a device that's actually pretty common in microbiology labs, and there's a picture of someone using it right below. When we make a spread plate, and again, you're going to see colonies growing all over a spread plate like this. When we make one of these spread plates in the laboratory, we can certainly count colonies by hand, and we do that all the time. But you can imagine that in some labs, they have so many spread plates to count every day that counting them manually is just not efficient. So, what they do instead is use semi automated and fully automated systems to help in that process. So, let's take a look at this machine first, this one here. Essentially, it's got this well cut into it with a bright light and a grid. You can see the lines of the grid right here. You put the plate here with the colonies on it. And what the person who's doing the counting has this stylus that is connected to this machine. And what all the person has to do is tap the stylus onto each colony, just tap it down, and that will get recorded as a count. So tap, 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 tap across the plate, and you'll get a count of how many colonies are on that plate, colony forming units again. You don't have to count, you don't have to tap on every single colony, by the way. Because spread plates spread colonies out nice and evenly across the plate or relatively evenly, you can divide the plate into sections, either using this grid or perhaps just dividing it into 4 places, 4 quadrants. And you can count what's in one quadrant and multiply it by 4. Again, it's just an estimate. It's an estimate. But you don't have to count every single colony on the plate perfectly accurately to make this work. So this stylus and this counting machine can make it a lot easier than doing it manually. And the last thing you can use is a fully automated system. And that's actually where this picture was generated. These are software systems. You take the plate that has the colonies growing on it, and you put it underneath this imager, this camera, essentially. It takes an image of the plate, and the software actually does the colony counting for you. So that's a fully automated system. And you can imagine you could count hundreds of plates using this computerized system in a single day. But automated systems, computer automated counting systems are pretty expensive. The software is pretty expensive. So unless it's a laboratory that does a lot of counting of colonies, you don't see it very often. Most of the laboratories that I've been in my working life, people counted colonies by hand. It's not hard. You get good at it pretty quickly. So that tends to be what I've seen most often. So what about the Rhodac plates? I said a minute ago that the Rhodac plates are going to pick up individual cells. Sometimes, if it's a particularly dirty environment, you could pick up enough cells so that you just got sort of a big clump growing on the plate. But more often than not, you're just going to pick up individual cells on the Rhodac plate, and they're going to grow into colonies overnight. So the next day, what you do is you count those colonies on each Rhodac plate, and you compare the colony count to a standard reference table to give you an idea of whether or not that count is good or bad. So this is a system that is not telling me what is growing. It's just telling me how many things are in that environment. So, for example, the Rhodac company itself puts out a reference table to use with these plates, and they describe 3 categories of microbe numbers in the environment. If you, for example, find 0 to 25 colonies growing on a plate, that's considered good. Between 26 and 50 colonies growing on a plate, that's considered fair. Anything over 50, that's considered poor. And you use these plates again to evaluate how well your facility is being cleaned and disinfected. A lot of institutions, though, a lot of hospitals, a lot of long-term care facilities that use these plates, they actually set their own guidelines. And they, in other words, they're stricter than the reference tables are. So take a look at this slide. This is one of the reference tables I'm talking about that can come with the Rhodac plates. When you buy these plates, the company sends you a standard reference table so you can evaluate your results, basically. So over here on the far left column, you can see what are called the critical areas. So for example, they've got floors listed for both the operating room, the obstetrics and gynecology wing, the nursery. They've got tabletops listed, patient rooms, floors and tables in those rooms, the bathrooms, the floors, the sinks, the toilet seats, other floors in the facility, other surfaces in the facility. All of these different places that you might want to sample in a hospital setting, for example, or a long-term care setting. So you can see these are the results that would be in the good category. These are the fair and these are the poor. So let's say, for example, it was your job, you were working in a hospital, and it was part of your duties once a month to, say, gather some samples from the nursery. And you went around the nursery and you pressed these Rhodac plates all over the floors in the nursery and incubated those plates overnight, and then got, say, an average count on your plates. And let's say you found 3 colonies growing per plate. That would give you an indication that your disinfection program is pretty good, because between 0 and 5 colony forming units on a plate is considered good. Let's say it was your job to sample in the bathroom of certain patient rooms. Let's say you had a patient in the hospital who had a particularly infectious, dangerous bacterial disease. And after that patient left, the room was disinfected, and then you had to go in and sample to make sure that the room was clean. So you went in and you took plates from the floor and the sink and the tub and the toilet seat in the bathroom. And let's say you found like 30 colonies per plate. That's going to put you over here in the poor category. I can't say for sure what is growing, right? Because I haven't done any testing to figure out what pathogen there might be. It might just be an environmental microbe that's had nothing to do with that patient. But regardless, there are too many colony forming units on that plate right now. And that's a sign that that room has not been disinfected correctly. If I want to know if those colonies are from the pathogen that that patient had that last stayed in this room, I'm going to have to do a lot more testing. I'm going to have to take those colonies into the microbiology lab, and I'm going to have to grow them in culture, and I'm going to have to do a lot more diagnostic testing to identify the pathogen. But the Rhodac plate will tell me how an estimate of how many cells there are in that room. So just to summarize a little of what we've been talking about, we refer to colonies when we're counting them on plates, whether they are spread plates or Rhodac plates. We call them colony forming units because we recognize that at the bottom of every colony, there was a parent cell. So this is essentially a cell count, but it is an estimate of the cell count. It's not a direct quantitative count. We can count colony forming units in liquid samples very accurately using spread plates again, or again, Rhodac plates. We can sample surfaces in the environment with these Rhodac plates with swabs, sterile swabs. We can count CFUs, but we just have to remember that if we're sampling surfaces, we're only getting data about that surface. So if I'm in a patient room and I'm sampling the bathroom, I can't make any conclusions about the rest of that patient room unless I sample other places in the patient room. You can only draw conclusions about the specific surfaces that you sample with swabs or Rhodac plates. So Rhodac testing specifically and using these swabs specifically, it's limited. It doesn't tell us precisely everything that's growing in the room. It doesn't tell us whether the entire room is clean. It gives us an estimate. It gives us an idea. If I want to get the best amount of information I can get about a particular room in a particular facility, I need to get a lot of swabs. I need to get a lot of Rhodac plates. I need to get a representative sampling of that room. If I just sample one area, all I can conclude is about that one area. So we're always taking multiple samples when we're doing this kind of testing. All right, we're going to switch gears now, and we're going to spend a few minutes talking about culture media. Remember, we've talked a little bit about things like the nutrient broth that we use in test tubes to grow microbes. We've talked about the solid agar plates that we use to grow microbes. We're going to talk a little bit more now about how media varies, because we do have many different types of culture media available to us in the microbiology lab. So culture media is specifically designed to grow or culture microbes, microbes that we've collected from the environment, for example, microbes that come from patient samples, microbes that come from any of a number of places. We place the cells into the culture media or onto the culture media with the direct purpose of trying to grow them. And in order to be successful in that, we have to add the things that those cells need to grow. So we have to have water in that media. We have to have some kind of a carbon source. And the carbon is essentially the skeleton of all of the different molecules that microbial cells synthesize every day. And generally, when we're talking about a carbon source, we're talking about protein. So proteins are added to culture media. There's also a material that's called peptone. All peptone is partially digested protein. So they take proteins, which can be very large. They can contain hundreds and thousands of amino acids in them, and they just hydrolyze it or break it apart a little bit to create peptone. Culture media needs an energy source. Remember, living things on our planet anyway, you tend to use carbohydrate for energy. Things like glucose get broken apart in order to make ATP for energy. And finally, microbes have requirements for certain vitamins and minerals. Those will also have to be added in. So we've got to give microbes what they need to grow, to survive and divide in culture. Now, all culture media have to have this minimum amount of nutrients in them, but they don't have to stop here. When we have this sort of limited list of ingredients, we often refer to this as a chemically defined culture media. In other words, we know exactly what's in it. We know exactly what kind of protein, what kind of carbohydrate, what specific minerals, what specific vitamins went into the media. So it is chemically defined media. But sometimes we add additional ingredients specifically to coax microbes to grow. Not all microbes are easy to grow. Some of them will grow on virtually any media, but other microbes, they're just harder to grow. They have more specific nutritional requirements. So sometimes we will develop what we call complex culture media by taking the basics and adding ingredients that are very rich in nutritional molecules, things like blood, things like egg and milk are nutritionally very dense. Meat extracts is another good example. Um, these types of ingredients, turn a defined medium into a complex medium. Gets the name complex because the truth is we don't know every single molecule that we have now put into that medium. We don't know every single molecule that's in blood. We don't know every single molecule that's in an egg, for example, or in milk. We know a lot about those materials, but we cannot say for sure exactly what's in the media when we start adding complex ingredients to it. So we refer to it as a complex media. And generally, when we use complex media, we're trying to grow an organism that we call fastidious. Fastidious means difficult to grow, has a lot of very particular nutritional requirements that you just don't find in ordinary chemically defined media. So we have it available to us when we are specifically trying to grow organisms that are a little harder to grow in the lab. Now, there are other kinds of culture media besides chemically defined and complex, and we're going to talk about those in a later laboratory. But for today, I just want you to be familiar with these two sort of categories of culture media. There are many different chemically defined media on the market, commercially available to us. And there are many different complex media available to us as well. So we can pick and choose the culture media that we think will allow us to be the most successful at growing the microbes we're trying to grow. Now, I also want to say a few words about Auger in terms of its role in culture media. There are, as I said a moment ago, there are liquid media that we refer to as broth. There are solid media that are based on this stuff called Auger. And then there are semi-solid culture media that are somewhere in between those other two. When we talk about Auger, or we talk about an Auger plate, the auger itself is just the base material. It's what is going to make the rest of the media solid. So auger itself is just a carbohydrate, but it's a type of carbohydrate that comes from a particular kind of algae that live in the ocean. These are called red algae, and they live in marine environments. and they use agar as their cell wall material. So by harvesting these algae, we can gather the agar out of all of the cell walls, and we can use it as a base to make solid culture media. So there's nothing in agar by itself that will help grow microbes. You have to add those basic ingredients that we mentioned a minute ago. You've got to add a carbon source, and you've got to add an energy source. The agar itself, even though it's a carbohydrate, is not the kind of carbohydrate that microbial cells can use for food. It's just providing the solid base for this culture media. We have to add in all of the nutrients. Now, why do we use agar? Like, there are other things we could use as solid material, but agar turns out to be almost perfect for growing microbes on. And I've got a list of reasons why right here on the slide. Probably the most important thing is that it stays solid in the incubator. Remember, we're going to put these plates into a hot incubator. It's generally about 35 to 37 degrees centigrade. So we can't have the agar melting. We can't have our plates melting in the incubator. That's not going to help. And agar will stay solid in an incubator. You have to get it all the way up to 100 centigrade to melt it. Now you can melt it, and we do all the time. We melt it so that we can pour it into plates, and we melt it so that we can add ingredients to it. But you have to get it up to 100 degrees centigrade to melt it. Once it is melted and you pour it into plates or other containers, you're going to get that nice smooth surface that is just right for growing colonies on. It's very easy to add nutrients to it. It's not hard at all. And as I said a minute ago, the vast majority of microbes will not digest agar. They won't use agar itself as a food source. They'll use the nutrients we put in the agar as a food source, but they won't degrade the actual agar. Of course, if they did, that would be a problem, right? If they ate the agar that we spread them on, that would be a problem for us. So it really does. It makes a very nice, very practical material for growing microbes on. You can buy agar in a granulated form, and then you just mix it up in the laboratory. You melt it. We generally just melt it in a microwave oven. It's very easy. Add the nutrients to it that you want to add, depending on which type of culture media you're making. And then you just pour it into sterile Petri dishes and let the agar solidify. It's actually a very easy process. It's not difficult at all to make these agar plates. All right, just one sec. Sorry, I've got a dog who is trying to get in my lap. She gets a little bit jealous when I teach a lab. She wants to get in my lap, and she weighs 50 pounds, so I have to discourage that at all times. One of the most common ways that we use agar is by pouring it in a plate like this and letting it solidify. So this is a container of solid material that we can grow microbes on. We can get colonies to grow. And this is generally the most common way we use agar in a Petri dish. But we can also put agar into test tubes for certain uses, and that's what you're seeing here. This image right here shows what we call Auger slants. Here's the word right here. What you do is you pour the liquid agar into these test tubes, and then you tilt the tubes while the agar is solidifying, while it's cooling, and you end up creating this. you end up creating a slanted surface of the auger. Now, why would you want to use auger this way? Well, sometimes you don't need colonies to grow on an auger plate. Sometimes you just need to get the cells to grow. Maybe you're going to do a metabolic test, or maybe you're going to grow them and then make another culture container, make a broth tube or something. So sometimes you can just smear a few cells onto an auger slant and just get the cells to grow. That's primarily why we would use them. Now, this tube over here, this picture is of what we call an auger deep. This time you pour the liquid auger into the test tube and you keep the tube straight up and down while the auger cools off and solidifies. And what you would use this for is if you had an organism that you needed to grow in an environment that had little or no oxygen in it. There are lots of microbes that find oxygen toxic. So what we do is we place some cells down deep in the auger, far away from where the air is. And they'll be able to grow down here because there's very little oxygen at the bottom of this tube. This is what we call an auger deep. In order to inoculate this tube, you don't use an inoculating loop. You use what's called an inoculating wire. Basically, what you do is you take a metal inoculating loop and you cut the loop off. So now you've just got a pointed wire. You dip the wire into your broth culture and then you stab the wire down into the tube. Remember, this is agar, so it's solid, but it's soft, like a gelatin. Down here on the bottom of the slide, you can see a couple of these agar deeps that have been inoculated and then incubated. So you can see the stab line right here. And you can see the stab line a little more blurry over here. So this is where that wire was just pushed down into the agar to leave some cells in the auger. Now, I think you can just make out that there's some kind of white material in here growing. These are bacterial cells growing in here. Over here, we also have white material growing, but look at how wide the line is. In fact, the auger itself has become very cloudy. That's because what we ended up putting into this auger deep was a motile cell. So we stabbed the auger, and those cells literally traveled out into the auger and started growing. So we use deeps to provide a particular area for oxygen sensitive organisms to grow. And we inoculate using a wire. The slants we're going to use just to give us a place to grow cells. We let the auger solidify on a slanted angle because we want that surface area. But we're not specifically trying to grow colonies here. We just need to get some cells to grow so that we can do something else with them. So agar is a really versatile material that we use in the microbiology lab. Again, most of the time we're using auger plates. They've got a nice big surface area. We can make quadrant streak plates. And remember, we do that because we want to get isolated individual colonies to grow. And we want them because we want to evaluate their morphology. We can also use hockey sticks to make spread plates. And we use the spread plates to count colonies so that we can calculate colony forming units as an estimate of cell number in a particular sample. So the plated auger is the most common way that we're going to use it. but we can also put it in a test tube and make use of it for other purposes. All right, so the remaining slides in this set are about colony morphology. I'm going to finish up today with this slide, but the rest of the slides in here, and there aren't that many, there are 4 or 5 slides. I've actually created a short video for you and put it up on YouTube in our playlist, so that you have that little isolated video that's just about colony morphology and some of the basics of colony morphology. So you'll want to take a look at that video when you have a moment, and learn just a little bit about colony morphology. But let's finish up today by looking at this slide. I just wanted to say a couple of words about this. I said a minute ago that the most common use for Auger in the microbiology lab is to make these plates. Well, these plates, there are 2 Auger plates here, and I think you can see that there's this yellowish sort of white material growing on each one of these, but it's growing not as colonies, it's growing as one big solid mass of material. This is what we call a lawn, L-A-W-N, a lawn of cells. Sometimes we do this on purpose on a plate. The way you get a whole, like, continuous lawn of cells to grow like this is by putting a lot of cells onto this plate. So we purposefully put a large number of cells onto the agar plate, spread them around to cover the whole surface, and then let them grow. Now, you might ask yourself, why would you need to grow a lawn? And the answer is sometimes we use lawns to test the sensitivity of a microbe to a chemical, something like an antibiotic, for example. You may have heard of what's called culture and sensitivity testing that we do when people get bacterial infections. If you look at this plate over here, this little white circle in the middle of the plate is a paper disc that has been soaked in an antibiotic solution. So what we did was we spread cells all over a plate to make a lawn again, but we also put this antibiotic disc right in the middle of the plate. Then we put the plate in the incubator and we let it grow for 24 hours. When you take the plate out, sometimes you see this, you see a clear zone around the disc. What has happened is all of these regions out here, the cells were able to grow into a lawn. But in the region that's right up near the antibiotic disc, none of the cells were able to grow. That's why you have the clear area. And what this tells us is that these particular cells are sensitive to this particular antibiotic. You have to measure these zones and you have to compare them to reference tables in order to say for sure whether or not you have an organism that is sensitive to an antibiotic. But this is how we do antibiotic sensitivity testing. We buy these commercially available paper discs that have antibiotic in them, and we just place them onto these plates that have been spread over with bacteria. Then we let it incubate. And if we get a clear zone around the disc, we have an idea at least that that organism was sensitive to that antibiotic and was either killed outright or prevented from dividing anyway. That clear zone shows us that those cells were sensitive to that drug and they weren't able to grow into a nice lawn like the rest of the plate has. So again, you've got this additional video to watch when you have a minute about colony morphology. And remember, all that word means to us is what a colony looks like. What does it look like? How big is it? What color is it? What kind of margin does it have? What kind of shape does it have? Does it grow above the auger? Does it stay flat on the auger? All kinds of characteristics like that can be used to describe the colony morphology for a type of bacteria. So that's available for you up on our playlist. That's all I have for you today. There are lab questions up for you this week that are due Sunday. You should be able to answer those now. I think you should be all set. And I do want to apologize for the couple of you that had trouble getting into the meeting today. Just for future reference, when you go into Canvas and you go into our homepage, when you click on where it says Zoom, you'll come straight into a list of our meetings. I also put some information on our homepage, which I think some of you tried to use today to get into the meeting, but sometimes that meeting information changes. I think I shared with you that I had a whole bunch of meetings disappear on Zoom. I had to reschedule. So the best, safest way, the most effective way to find our lab meetings is not to go into Zoom directly and enter meeting information, but to click on where it says Zoom in Canvas and look for the scheduled meeting and then click where it says join. So again, I apologize if you had trouble finding it today. Unfortunately, we have problems with Zoom from time to time, and we just have to try to adapt as best we can. So that's all I have for you today. If anybody has any questions about anything we've covered, I'm happy to answer those. If not, enjoy the rest of the evening and have a good weekend. And I will see you again next week.
Thank you.
You're very welcome. We'll see you next time.